Hi, in the next two lectures, we're going to continue our discussion about functional association networks. We're going to learn how to create functional association networks from data, as well as analyze gene lists using functional association networks to build subnetworks that connect lists of genes using various data types. So this is a refresher from the previous lecture. Here, Tanai et al. Created a bipartite graph that links genes based on various functional properties. They use this approach to identify functional modules in the Yeast regulatory network. So here I'm showing you for the first time a concept that will be repeated throughout the lectures. This is a gene set library, or an example of a gene set library. In this text file, opened in Excel here, the first column is a functional term. Here, for example, is the name of a pathway. On each row, there are a set of genes that share the functional term. For example, here they would belong to a specific pathway. So the gene set are members of the pathway. This file format was invented at the Broad Institute, and it's called Gene Matrix Transpose, or GMT file, and it was created for performing gene set enrichment analysis, GSEA. GSEA will be covered extensively later in the course. Here we will convert gene set libraries to functional association networks. So we have an inference problem. We're trying to infer a network from a gene set library. Given one or more GMT files or gene set libraries, can we infer the underlying unknown network based on co-occurrence of genes in sets from the library? We have to assume that there is an underlying network that connects those genes. And the information in the gene set library tells us something about the structure of this network. So we will first demonstrate how we can infer a network from partial information, and we show that this is possible on a synthetic network. On the left, we have a randomly connected network made of 10 nodes. Our goal is to infer the network given partial data. And in the center, we have an adjacency matrix representation of the same network. The rows and columns are the nodes, and there is an orange square if the nodes are connected. On the right, we have a small GMT file made of sets of various sizes. This library was created by performing short random walks on the example network on the left. This library could be the gene set library that we showed before. We just can replace the numbers with the genes. So now the question remains, can we infer the network using this small gene set library of numbers? Do we need more examples? How many examples are needed to fully infer the network? And what about large networks? So the answer is we can fully recover the network given enough examples. The algorithm Sets2Networks that was published in this paper in BMC Systems Biology creates random graphs that connect the nodes in a way that is consistent with the data from the GMT file. We create many possible networks like this, and the accumulated information about those networks inform us about the underlying real network. So while we're making those small networks that are consistent with the data, we keep account on how many times a link appears between all pairs of nodes. We then introduce a threshold to only keep those links that appear many times. Here you can see the network inference process live. On the top right is the network from the previous slide. We want to infer this network. On the top left is the matrix that represents the number of times a link appears between a pair of nodes in those small random network and are consistent with the data. The adjacency matrix in the center is the same as the one on the right, but here we applied a threshold to only include the most frequent interactions. The small networks that you see are randomly connected subnetworks that are consistent with the data. We need to create many of them to arrive at the correct solution or to fully recover the network topology. What we did next was to examine various parameters in this process. We wanted to know how many lines or how many sets we need to infer a network of a certain size. When we create this gene set library, are we going to use long or short random walks? Can we introduce errors in those random walks? And so on. To evaluate this mathematically or computationally, we use the Matthews correlation coefficient. This is a measure that is typically applied in the machine learning field, and it computes a balance between true positives, false positives, true negatives, and false negatives. We can think of each link in the network as a case and count the number of times correct and incorrect links were inferred. So here we have some results. So the Matthews correlation coefficient for attempting to infer a random network of 50 nodes using various size of gene sets of various lengths. If we have 180 of those short random walks on the network of 50 nodes, we can fully recover the network. The exactly correct network is inferred. There is a sweet spot of a threshold that we put in that if we just use that window of a threshold, we can have a full recovery of the exact correct network. Now we are getting to the fun part. We're going to apply this network inference algorithm to real data. So the first data sets that we're going to analyze using this network inference algorithm is the analysis of high throughput IPMS data. IPMS stands for Immuno-precipitation followed by mass spectrometry. So first, what is immuno-precipitation? Using a cell lysate, which means that the cells were broken up, the membrane was broken up to isolate either the cytosol or the nucleus. We mix the proteins from the lysate with an antibody. And an antibody is a small protein that is targeting a specific protein. It has a target protein that it can bind to. We then pool the antibody together with the target protein to isolate our protein of interest. However, the method does not always work perfectly. The antibody can bind to other proteins, and typically the target protein is also bound to other proteins that get pooled together with the antibody as a protein complex. Since we want to identify protein-protein interactions for our target protein or protein-protein interactions in general, this is not necessarily always a bad thing that the antibody binds to other proteins as well as the antibody pulling out many other proteins, as we're going to see later. After we isolated the complex that is bound to the antibody, we can identify the proteins in the complex using mass spectrometry proteomics. The isolated proteins are first digested into short amino acid fragments, and then those short amino acid fragments can be sequenced using a mass spectrometer. The resultant identified short sequences can then be aligned to the proteome or the genome databases to identify the proteins within these complexes. So we cannot apply our network inference problem to one IPMS experiment. But if there is data about many such experiments using different baits in a similar system, we can consider the protein list resulting from such experiments as the sets in our gene set library, and then use the method to infer direct protein-protein interactions. Luckily, A paper in Cell was published where the authors did over 3,000 of such IPMS experiments in breast cancer cell lines targeting nuclear receptors and other membranes of the transcriptional machinery. That project took 10 years to complete. So this is a great dataset for applying our network inference method because we can validate our predictions with known protein-protein interactions previously published using low-throughput methods to identify protein interactions. So here is a visual comparison of the adjacency matrices of the predicted protein-protein interactions for the 3,000 proteins that were detected in the high-throughput IPMS study on the left, and their previously known protein-protein interactions from low-throughput studies on the right. You can see that the method recovers many of the known complexes and binary interactions that were previously detected by low-throughput studies, as well as predict likely new undiscovered interactions. So just visualizing the results of the predictions next to the known interactions is not a proof that the method actually predicts known interactions. To show that the method predicts real interactions better than randomly, here we plot the false positive rate versus the true positive rate as we change the threshold based on the likelihood score for the interactions using the Sets2Networks algorithm. You can see that the method predicts many known interactions for those protein pairs that have very high predicted interaction score. This type of plot is called a ROC curve, and it is also very commonly used in machine learning for evaluating binary classifiers. The second application of the method is from the new field of systems pharmacology that I promised to discuss. The FDA has a system to track the side effects of drugs after they are approved to potentially identify toxic drugs that should be withdrawn from the market. So doctors in the US are not required but encouraged, or they can volunteer to submit information about side effects or the adverse events that they face when treating their patients. They enter anonymous information that reports which drug a patient took and what side effect or adverse event those patients experienced. There are millions of such records, and they're available over a span of 10 years. Using this data as a gene set library, we can infer interactions between drugs and between side effects and between drugs and side effects. So here is an example of the mean adjacency matrix that results in applying the Sets2Networks network inference method to commonly used cancer drugs and 40 severe side effects. The matrix identifies drugs that are co-prescribed, side effects that co-occur, and likely side effects for drugs by looking at the bright spots on the rectangles on the top right and bottom left. And here is an application of the method to 1 million records from this FDA adverse event reporting system for all side effects and drugs. We can zoom in to see interesting regions where we find interactions between drugs and side effects. And the final application is to generate a scientific collaboration network. We can consider publications as lines in a gene set library or in a GMT file, where the authors are the genes and their co-appearance in publication is an indicator of their level of collaboration. So in this network, you can see Ravi, myself, and Eric Sobey, which is a zoom in to the Mount Sinai Collaborative Network. In the next lecture, we will explore how we can build functional association networks that connect lists of experimentally identified genes.